Right. Ladies and gentlemen, welcome to today's Comstec webinar on E. coli as a model organism. It is by Professor Dr. Ute Römling, who's a professor at Department of Microbiology, Tumor and Cell Biology, Karolinska Institute, Stockholm, Sweden. Before we commence with the webinar, I'll briefly go over our new flagship project, which is called the Comstec Technology and Innovation Portal. So I'll just share my screen and introduce you to our new project. So Comstec has launched a new project under the guidance of our Coordinator General, His Excellency Professor Dr. Mohammad Iqbal Chaudhry. It is called the Comstec Technology and Innovation Portal. It is basically a digital platform that is provided to the OIC countries to showcase their emerging and existing technologies. By doing so, we aim to provide a platform for academia industry linkages. We provide a platform for investors to look into. This is the basic outlook of our technology innovation portal. We are inviting exhibition in various fields of science and technology. These includes, as you can see here, information and communication technologies, medical health sciences, energy, and so more. Currently, it's still in its beta version, and in its initial phase right now, 31 companies have registered. By companies, it can be either an individual, it can be a company, it can be an institution, organization. The portal caters to all of them. So far, 15 products have been listed. There are 5 prototypes and the rest are commercial. So our portal basically caters for 2 types of individuals. One are those that want to display, they want to exhibit their technologies, they want to exhibit their products, services. These can be at the prototype level or at the commercial level. They can do so by simply signing up to the portal here through the sign-in option, and they get authorized within 24 to 48 hours. And afterwards, they can publicize their products or services. The second category is those that want to search for products. They can search for products depending on their need, whether they're looking for prototypes or commercial products in their area of interest, or whether they're looking for specific product services from a specific country or at a specific stage. Here you can look into specific products, go into their details, contact the individual or company or organization, and that's how our portal works. So we invite you all to visit our portal and register. You can also access this portal from our comstech.org website. We have a direct link from there as well, or technologyportal.comstech.org. With that, I would now like to invite our Coordinator General, His Excellency Professor Dr. Mohammad Iqbal Chaudhry, for the welcome address of this webinar.
Thank you, Asif. Bismillah ar-Rahman ar-Rahim. Good morning and good afternoon. We are in different time zones. Ute is still about 12 o'clock, but there are people from West Africa who are in the morning zone, and then we have many attendees from the Southeast Asia, which are already in the evening zone. I'd like to welcome you to this very important Distinguished Scholars program, in which we invite the chosen few, the best of best in their disciplines. And today we have Professor Dr. Ute Römling, who is not only a personal friend, but also a scientist of a very high caliber. She is known for very high quality science, and her capacity to link with many younger people in the developing world and help them in their career. So she has been truly an ambassador of science for global peace and development. And since yesterday we have celebrated United Nations Science Day for Peace and Development, I think Ute's lecture is more timely because she is from Germany, working in Karolinska, helping scholars in different regions of the world, including Pakistan. And this is the true spirit of science. Professor Römling is a very high caliber scientist, as I mentioned. She has been working on various infectious diseases and trying to understand how bacteria functions. For instance, her work on bacterium E. coli is one of the most well investigated due to its occurrence in almost every human, along with being one of the major pathogens for human. She has been working on E. coli and others. However, very little is known about E. coli as a commensal, despite the fact that E. coli K12 is a laboratory model organism and used in different laboratories for a variety of different kinds of microbial work. In this webinar, Professor Römling will analyze her research finding of a recent E. coli K12 clonal member in post-industrial human being. This is an extremely important seminar dealing with a very important topic. And let me have the pleasure of introducing Professor Römling. She is Professor of Medical Microbiology, Microbial Physiology at the world famous Karolinska Institute in Stockholm, Sweden. Her research focuses on molecular mechanism of biofilm formation, mainly in enterobacteria such as Salmonella, and persistence mechanism of Pseudomonas, worldwide spread, clone C. Professor Römling has over 129 publications, 43 peer-reviewed reviews, 2 peer-reviewed protocols, 10 popular scientific publications, 15 book chapters, and 1 book editorial. Among others, she has received research grants from the Swedish Research Council, ALF Foundation, European Commission, and German Research Foundation. In 2000, she has received the Young Investigator Award for research in clinical microbiology and infectious diseases from ESCMID, European Society of Clinical Microbiology and Infectious Diseases. Professor Römling has also been an associate faculty at the International Center for Chemical and Biological Sciences, University of Karachi. She's a member of the European Academy of Microbiology since 2017 and has been elected for Academia Net in 2016 and as a member of USERN since 2020. We're very fortunate to have someone of her caliber who is right at the frontier of this very important disciplines of biomedical sciences. And with this, I would like to invite Professor Ute Römling for her presentation.
Thank you, Professor Tutari, for this very nice presentation. I feel very honored that I can give this lecture in the context of Comstech. So my lecture will be on Escherichia coli as a model organism. Escherichia coli has been studied for a long time, but still we know very little about this bacterial species, as I would like to demonstrate you also in this lecture, as we know very little about biology in general. So but in particular, why do we still need microbiology, the study of microorganisms? Also, we thought at some time that we have almost eradicated all infectious diseases. However, antimicrobial resistance among bacteria is increasing with globally disseminating clones. Nosocomial hospital-acquired infections in the clinics in susceptible patients is also on the rise. And in general, also, we have a much higher living standard today. The population becomes more susceptible to infections. The population becomes older and requires more medical care. For example, in Sweden, has with 10% the highest rate of child diabetes in Europe. And the rate of diabetes in the Asian population can be up to 25%. diabetic individuals are more prone to infections, cancer, and other diseases. For example, in diabetic foot ulcer, a chronic wound involving bacteria infection, Pseudomonas aeruginosa, a biofilm-forming persistent pathogen, is frequently infecting. And now, for this lecture, important is Escherichia coli is a major cause of extraintestinal infections in patients with diabetes. Due to the high blood glucose level, this leads to more severe infections. But in general, why is there still a need to investigate the species Escherichia coli? Escherichia coli is found in the gut of almost every human being, as Professor Chutari already mentioned. It is one of the first colonizers of the human gastrointestinal tract after birth. It prepares for the colonization by anaerobic microorganisms. And Escherichia coli has also beneficial effects, such as the production of vitamins like vitamin K. But on the other hand, Escherichia coli is also a major cause of human intestinal and extraintestinal infections. The model strain that has been used in the laboratory since decades is Escherichia coli K12. Escherichia coli K12 was isolated in 1921 in Stanford from a human being reconvalescent for diphtheria. This strain has been found to be very closely related to NCTC86. This is a strain originally isolated by Theodor Escherich in 1886. But this E. coli strain has gone through manipulation steps and several derivatives are available. The model organism, which refers to a very original version of this strain, is MG1655. Thus, we postulated that a modern E. coli K12 strain would be desirable to investigate the adaptation to post-industrial human beings. Escherichia coli is a highly diverse species. As you can see here in this phylogenetic tree, it is divided in different phylogroups. We were interested to collect a number of commensal E. coli strains in order to investigate their characteristics. And what we could see is that we could isolate one human strain that is closely related to Escherichia coli K12, the model strain that has been used for decades. This is shown in even a much more detailed phylogenetic tree, where all fully sequenced Escherichia coli genomes are displayed. In red, you can see E. coli FEC10, this human commensal strain closely related to Escherichia coli K12. It was isolated from a healthy human being. It belongs basically to a certain sequence type 10. This is defined as the allele homology between different isolates. But you can also see in this tree that actually the species Escherichia coli is highly diverse. It is divided into several different phylogroups. And I only would like just to mention within the phylogroup P12, the ST sequence type 131 isolates, which are today a global problem as they ubiquitously spread, causing urinary tract infections, but also very successfully colonizing the human gastrointestinal tract and being present in the environment and in animals. What we found when we sequenced the genome of the commensal strain Escherichia coli FEC10 was that it contains a 156 kilobase pair plasmid. This plasmid has been derived from the phage P1, but it has been modified, so we expect that no functional phage can be made. However, the remnants of the phage and the scaffold of the plasmid is still highly phage related. We can also see that the origin of replication, where plasmid amplification starts, is actually the one that is present in the P1 phage genome. But the remarkable characteristics of this plasmid is that this plasmid encodes a lot of stress response genes. For example, the transmissible locus of stress tolerance, TLST. It also contains arsenate reduction operons. It contains a di-P decolorizing peroxidase, lactate utilization, and tetracyanate reduction operon. So the presence of these genes point to the fact that maybe this human organism has experienced a highly toxic environment. We were specifically interested in the transmissible locus of stress tolerance. This locus is approximately 20 kilobase pairs long, and it contains a number of conserved genes. Remarkable on this transmissible locus of stress tolerance is that it does not contain antimicrobial resistance cassettes or virulence genes, but it contains genes involved in, for example, protein homeostasis, in cell envelope stress, and in oxidative stress. It is present not only in Escherichia coli, as on the plasmid that I showed you, but it is also present, for example, in Pseudomonas aeruginosa. So what does these do, the genes that are present on this plasmid do when they are translated into proteins? We know that protein aggregation actually contributes significantly to aging. It contributes to aging not only in human organism, but it contributes significantly to aging also in bacterial organisms. In Pseudomonas aeruginosa, where this diagram comes from, we have basically, by the presence of the TLST locus, a duplication of core genome functionalities. I just give two examples of this. On the core genome of almost any bacteria, you find the membrane-bound protease FTSH. This is a central proteinase on the core genome. The TLST locus contains a second copy of this core genome gene. This protease is involved in protein quality control, and we know at that time is that they have different types of activities, and they have maybe also different types of substrate specificities. However, I would like to raise your focus on these CLUB proteins that are displayed on the lower part of this slide. CLUB G is a disaggregase, and the disaggregases are usually involved in the renaturation of proteins. They take protein aggregates directly or indirectly and channel them into linear polypeptide chains in order to fold them again into native proteins. CLUB-G disaggregase is a central part of the stress tolerance island. And that we have shown in strains of Pseudomonas aeruginosa. When we have the native protein, we get to disordered aggregates, and this ordering of proteins can be prevented by so-called small heat shock proteins. However, when a protein eventually is disordered in an aggregate, then the disaggregase, like ClybG, is refolding and to a native protein. Bacterial cells which produce this type of proteins have a higher heat shock survival than bacteria that do not possess this type of disaggregase. And this we could show already also on the population level in Pseudomonas aeruginosa. Non-clone C strains have a much lower heat resistance than clone C strains which possess the ClybG disaggregase. This heat resistance is actually horizontally transferable, and strains which are usually more susceptible to heat upon acquisition of a 3-gene operon, DNA, small heat shock protein, and the ClybG disaggregase become more than 10 times higher resistant to higher temperature, to lethal heat exposure. However, back to Escherichia coli, when we screened the commensal strains that we had collected for high thermotolerance, we found that with 4 strains that possessed the transmissible locus of stress tolerance, and 3 of those isolates actually had a very high heat tolerance. Why did we do this screening? We did this screening because it was known before that the transmissible locus of thermotolerance is present actually in E. coli isolate derived from food. When cheese is fermented, a higher temperature arises, and this selects for heat tolerance isolates. So Escherichia coli found from fermented cheese and found also from beef samples contain often, due to the processing of those food products, a high number of thermotolerant strains. In contrast, the commensal isolates of Escherichia coli, which do not harbor the LST locus, show a much lower resistance to lethal heat shock. We could then also trace this high stress tolerance to the presence of the 3-gene operon, DNA, small heat shock protein, and the ClybG disaggregase. When we deleted this genetically with genetic manipulation, the disaggregase or the 3 genes, we can see that heat tolerance of the thermotolerant strain E. coli FEC10 diminishes dramatically. So the disaggregase ClybG is a major mediator of pronounced thermotolerance in commensal isolates of Escherichia coli. How does protein disaggregation work when we don't have the ClybG disaggregase, which is obviously horizontally transferred into certain Escherichia coli strain? The very well investigated and ubiquitously present disaggregase is the ClybB disaggregase. Thereby, in order to disaggregate the proteins, a protein aggregate has first to bind to the heat shock protein 70, also called DNAK. When these aggregates bind, they are provided to or presented to the disaggregase ClybB, and upon presenting an interaction, only then disaggregation can take place with linear polypeptide chains produced, release of the unfolded polypeptide chain, and either spontaneously or chaperone-induced refolding of the protein. The remarkable feature that we found with the CLUB-G disaggregase is that this disaggregase actually works without the aid of the co-chaperone DNA K. When we assessed heat aggregated malate dehydrogenase or heat aggregated luciferase, we can see that CLUB-G performs very well in refolding the denatured proteins. And this is shown in the graph with the red, in the red graph. Whereas when we want to denature, when we want to renature the protein with CLUB-B, we require also the co-chaperones DNA K, DNA J in order to get refolded protein. Otherwise, we have no activity when we use CLUB-B disaggregase alone. So this novel feature of this CLUB-G disaggregase shows we have a very efficient system to disaggregate proteins. However, what arises is, why does CLUB-G is not present in all Escherichia coli strain if it is such a efficient disaggregase? When we look at the disaggregase of CLUB-G from the commensal E. coli FEC10 strains and compare it to the disaggregase from Pseudomonas aeruginosa that we investigated previously, we could see that the disaggregase of the commensal E. coli strain has certain specific properties. Also, it is highly similar to the disaggregase from Pseudomonas aeruginosa. Still, we see a higher ATPase activity, and we see also that the susceptibility to a co-substrate casein is different. The disaggregation activity is also substantially different between the CLUB-G from commensal E. coli strains compared to Pseudomonas aeruginosa. This might be true. This might be relevant because E. coli strains have grow usually at higher temperatures than Pseudomonas aeruginosa strains and might need a more thermoresistant disaggregase. So we can ask in general, why do not all Escherichia coli or also Pseudomonas strains harbor this transmissible locus of stress tolerance? In general, on the average, only 2% of Escherichia coli isolates harbor this locus. And when we go to Pseudomonas aeruginosa, we have a prevalence of approximately 60% in environmental isolate and 20% in clinical isolates. So there might be a disadvantage of this transmissible locus of stress tolerance under certain environmental conditions. Or there might be a disadvantage in certain disease situations. There might also be that not all the genetic backgrounds of the bacteria are favorable for the presence of this transmissible locus of stress tolerance. In interaction with the human host, there might be an immunological interference. This is at the present an unknown biological activity in host interactions. However, in the bacterial group B disaggregase has been shown to be a conformational antigen mimetic of the anorexigenic peptide alpha MSH and is at the origin of eating disorders. I will now switch to another topic within commensal Escherichia coli strains that we have investigated, and that is biofilm formation. What we have observed over the years is that we have a diversity of biofilm formation in Escherichia coli. And we look at a specific type of biofilm formation that is called the radar morphotype, and radar stands for red, dry, and rough. And this biofilm type has been named according to the appearance of these colonies on agar plates that contain the dye congoret. What we can see is that cells expressing radar morphotype express actually two extracellular matrix components, cellulose and curli fimbria. And upon expression of only curli amyloid fimbria or upon the expression of only cellulose, we see distinct biofilm phenotypes on agar plates. This is a very simple but convenient and relevant model to assess one type of biofilm formation in Escherichia coli strains. What we have observed is when these Escherichia coli performs this type of biofilm, we can see different interaction with the host cell. In the case of the full radar morphotype, we see biofilm formation in interaction with the cell line HT29, while when we delete this biofilm formation, we see only single cells attaching to the surface. When we delete one of the extracellular matrix component, either cellulose or curli fimbria, we see differential adherence of cell aggregates to the surface of epithelial cells. And this surface behavior is then also extrapolated into internalization upon matrix production of cellulose. We see even a prevention of internalization, while upon matrix production of curli fimbria, we see elevated internalization compared to the wild type into the intestinal epithelial cells. However, when we have no biofilm formation, we see even less interaction and internalization. But what we can conclude in comparison to the wild type here is that we have basically two extracellular matrix components in the bacteria expressed, but those extracellular matrix components have actually opposite functionality. One of the matrix components is promoting invasion and internalization, and one of the extracellular matrix component is basically inhibiting the internalization into host cells. We have a similar pattern of opposite functionality with respect to induction of the pro-inflammatory cytokine IL-8. The wild-type strain shows the intermediate production or triggers an intermediate production of IL-8 from intestinal epithelial cells, while cellulose alone is inhibiting and curli expression alone is is triggering this type of interaction with the host cell. So again, we see two types of opposite function. We see opposite functionality of these two consecutively expressed extracellular matrix components. This pattern extends also to virulence and immune induction of an urinary tract infection isolate of Escherichia coli. We see upon cellulose expression a much lower kidney colonization as when we have coli expressed alone or when we have a full morphotype. Similarly, we see upon that cellulose expression in urinary infection isolates in Escherichia coli strain 12 delays neutrophil-mediated bacterial elimination in vivo. So the interaction with the host cells is, so to say, less. We could also show that expression of the biofilm components protects the urinary tract infection isolate against the antimicrobial peptide LL37. And this is shown here. So when we have coli fimbria alone, we can see protection as with the wild type, while when we express only cellulose or when we express none of the extracellular matrix components, we have a much higher susceptibility to the antimicrobial peptide LL37. But there is a dual interaction, as the antimicrobial peptide LL37 inhibits also biofilm formation of Escherichia coli strain K12. And the reduction in biofilm formation can be quite substantial. However, when we use a scrambled peptide, we see no reduction of biofilm formation. And also, when we take an unrelated antimicrobial peptide, we see no reduction of biofilm formation. So this reduction of biofilm formation is quite specific to LL37 and its interaction with RADA-producing Escherichia coli. We could then show that specifically the LL37 binds to the monomeric coli subunit CSGA in order to prevent polymerization. However, we know also that this is not the only mechanism how LL37 prevents biofilm formation as an innate immune response against biofilm forming bacteria. In conclusion here, the expression of coli fimbria by the uropathogenic strain E. coli K12 protects against the bactericidal activity of the antimicrobial peptide LL37. Expression of coli fimbria and cellulose protects against immune components of the bladder epithelial cell line T24. The antimicrobial peptide LL37 prevents biofilm formation of Escherichia coli at sub-inhibitory concentration. And the antimicrobial peptide LL37 binds directly to CSGA, the monomeric subunit of coli fimbria, and thereby preventing polymerization of CSGA into amyloid coli fibers. As a third part of the diverse aspects of Escherichia coli, I would like to discuss the impact of horizontal transfer of a novel cyclic dinucleotide signaling gene into Escherichia coli. what we have seen at some time point in a animal commensal isolate of Escherichia coli EcoR31 is that we have on the right border of the high pathogenicity island, we have a signaling module. And this signaling module consists of a number of genes. One of the genes is called, encodes for a dinucleotide cyclase. This gene and the protein product have been previously characterized to produce cyclic AMP GMP. However, it is otherwise not known that Escherichia coli is producing cyclic GMP AMP. So we were wondering what is happening when a novel second messenger signaling system is actually introduced into a species. What we have shown before is that the ubiquitous second messenger cyclic GMP is a global regulator of satiety, motility, and chronic acute infection lifestyle switch. Upon high cyclic GMP level, we promote biofilm formation and chronic virulence, and on the single cell level, satiety. While upon low cyclic GMP levels, we promote motility and acute infections. This type of lifestyle switch is regulated by cyclic GMP is not only present in Escherichia coli, it has been shown also for Salmonella typhimurium, Pseudomonas aeruginosa, and other bacterial species where it has been investigated. So we were wondering first whether the cyclic GMP signaling system in the commensal strain E. coli EcoR31 is functioning in the same way as it has been shown for other bacteria strains and species. So we overexpressed a digranulate cyclase, and upon overexpression of the digranulate cyclase, we see higher biofilm formation. While upon overexpression of a phosphodiesterase, which is downregulating, hydrolyzing the second messenger cyclic GMP, we see a downregulation of biofilm formation as indicated by a white colony phenotype. On the other hand, expression of a digranulate cyclase is downregulating motility, and expression of a phosphodiesterase is upregulating motility. So the cyclic GMP signaling system and its impact on bacterial lifestyle behavior is not affected by the introduction of a novel second messenger signaling system. What we could show, however, is then that DNCV, that dinucleotide cyclase, which produces cyclic GMP AMP, is also involved in the regulation of biofilm formation and motility. However, in a different way, it downregulates both biofilm formation and motility. In this, by investigating this, we came to this type of regulatory regime. We have the new dinucleotide cyclase DNCV, which is producing not only cyclic GMP AMP, it's a more promiscuous dinucleotide cyclase. It is producing also other cyclic dinucleotides in vivo and in vitro. And the production of the cyclic GMP AMP most likely inhibits biofilm formation, and as it inhibits motility. We were then wondering what is a receptor that is involved in this type of regulation. And we come only to the first conclusion is upstream of the gene for the cyclase is a gene encoding for a patatin-like phospholipase. And this phospholipase has been shown to be a receptor for cyclic GMP-AMP in Vibrio cholerae. However, how should a phospholipase regulate biofilm formation and motility? This is still an obstacle. However, what our attention was a little bit drawn from the subjects, as we have found another subject with this cyclic GMP-AMP receptor that I call evolution of the patatin-like phospholipase. And what you can see here on this display is actually Escherichia coli K12 cells that have been elongated, and the elongation is dependent on a variant of the patatin-like phospholipase CAPV, which is the cyclic GMP-AMP receptor. So what we would first ask is, of course, when we characterize a novel second messenger signaling system, what are the mechanisms of cyclic GMP-AMP to downregulate motility and biofilm formation? What is the identity of the receptors? Which enzymes degrade cyclic AMP-GMP? And are there alternative targets beside the biofilm regulator, CSGD, and motility? And the first, of course, subject is to look at the already known receptor for cyclic AMP-GMP, the patatin-like phospholipase, DNCV. So shortly, what is a phospholipase? A phospholipase is an enzyme that hydrolyzes phospholipids, the major constituents of a lipid bilayer membrane. And phospholipases are classified according to the cleavage site within the phospholipid molecule. They can cleave at the different ester bonds, and they are then designated PLA-A1, PLA-2, or PLA-C and PLA-D. And patatin-like phospholipases are a subclass within the PLA-2 phospholipases. As a hydrolytic activity that targets the membrane of bacterial cells and also, of course, of eukaryotic cells is highly detrimental, all phospholipase enzymes need to be highly regulated, and their catalytic activity needs to be stimulated. So they are basically, without stimulation, they are not catalytically active. Traditionally, there is a discrimination between a calcium-dependent and a calcium-independent enzymes. These patatin-like phospholipases are defined over the conserved patatin-like domain fold with conserved consensus residues, including a serine hydrolase motif and the active aspartate. Patatin-like phospholipases include, but are not restricted to, these calcium-dependent and calcium-independent enzymes. Importantly, besides remodeling of the membrane, patatin-like phospholipases have also signaling functions, as they produce lysophospholipid and fatty acids, which both can be also signaling molecules. Patatin-like phospholipases of bacteria are poorly described and have not been classified. Besides maybe one type of patatin-like phospholipases in bacteria that are virulence factors. And those virulence factors, upon injections into host cells, are activated by host factor. A prominent example is the XOU patatin-like phospholipase, a virulence factor of Pseudomonas aeruginosa that is secreted by the type 3 secretion system and can be activated by monoubiquitination or polyubiquitination. But what we accidentally found actually when we looked at the biological activity of individual components of the DNCV signaling module is that we accidentally isolated a variant of the patatin-like phospholipase CAPV, and this variant of the patatin-like phospholipase CAPV Q329R downregulated motility independently of DNCV, while the wild type protein did not have any effect. We were wondering then what is a reason for this inhibition of motility on the single cell level. And when we looked a little bit closer, when we express either the wild type patatin-like phospholipase or the variant phospholipase, is that we could see that the variant phospholipase actually led to an extension of the cells into very long filaments, while the wild type patatin-like phospholipase has only a very minor effect on the cell length. This effect holds for us a long time, but upon depletion of the induction, the cells revert to single cells again. So this is a reversible process. It's not only a stress response, as we can see it, for example, when we expose bacterial cells to certain antibiotics. We can also look at this cell elongation in more detail on the single cell level. And what we can see is that upon expression of the variant CAPV protein, we see not only inhibition of cell division, but we also see nucleoid elongation. As we have patatin-like phospholipase, and we were wondering whether this variant patatin-like phospholipase has a higher cytotoxicity as the wild-type protein. We looked at the cell growth, and we looked at the colony-forming units at the viable cells upon extensive expression of these proteins. And we could see that we see a reduction in viability. Also, the reduction in viability is not substantial. It is only approximately twofold. So it might be that the enzymatic activity might be enhanced upon the Q329R substitution in the protein. We were then wondering whether this is a constitutive feature of the patatin-like phospholipase CAPV. However, we find amino acid substitutions that can relieve the motility and filament motility inhibition and cell division inhibition. But this biological activity of the variant CAPV Q329R is not constitutive. It is prone to be affected by environmental conditions. With vitamin B12, vitamin B6, for example, substantially inhibits filament production by CAP-329R. Other effects of the patatin-like phospholipase are, for example, a downregulation of other biofilm formation, which does not occur upon overexpression of the wild-type protein. We were then wondering whether this effect of the patatin-like phospholipase CAP-329R is restricted to this specific Escherichia coli strain that comes from an animal, EcoR31. However, upon overexpression of the variant protein in a number of Escherichia coli strains of commensal, but also of pathogenic origin, we can see that filamentation is almost ubiquitously triggered by the patatin-like phospholipase. Also, there are some exceptions, like this commensal strain FEC89, which shows only a very slight elongation of the cell length upon expression of the patatin-like phospholipase CAP-V. But also other species like Salmonella typhimurium show this phenotype. Here also we see extensive cell filamentation. What we think is that, of course, one might think that this behavior of the patatin-like phospholipase is an arbitrary behavior. However, we think that this is a highly relevant biological behavior that can maybe also explain some biological features of patatin-like phospholipases in humans. So in conclusion, when a cell divides, we first have the chromosomal replication. We then have a cell elongation concomitant with nucleoid segregation. As upon successful nucleoid segregation, we see invagination of the cell membrane and a so-called Z ring formation, which is the initiation for cell division. And we also have nucleoid condensation. And upon successful invagination, we have the division of one cells into two daughter cells. When we overexpress the patatin-like phospholipase CAP-V Q329R variant, we see an extended elongation of the nucleoid. And we see also an extended elongation of the cells due to restricted cell division. There is also insufficient nucleoid segregation. And sometimes, but not in all occasions, no nucleoid condensation. We see invagination, but the invagination is reduced equally as the Z ring formation is reduced. So we have a substantial number, or the majority of the cells are long filaments where cell division has been inhibited. Concomitant with inhibition of cell division, we observe a downregulation of biofilm formation, and we also observe a downregulation of the motility that is due to the absence of polymerized flagella on the cell surface. With this accidentally discovered variant of a patatin-like phospholipase, we have, through one amino acid substitution, created a protein which has a quantum leap functionality change. What is the molecular mechanisms of this quantitative functionality change, however, needs still to be investigated. It can be that the polymerization of the enzyme is inhibited or is altered. It can be that the enzymatic activity of the enzyme is altered, or the substrate specificity is altered. Also, we see at the very first glance, without going into molecular detail, no really significant hints for such a behavior. So this remains to be shown in future studies. So last but not least, I would like to thank you for your attention, and I would like to acknowledge all the people in my research group and all the collaborators which have been involved in this work. Without them, this work would not have been possible. So thank you for your attention.
Shall we take questions?
Yes, please.
What I'll do is all the participants, whoever has questions, just type them down in the chat, and then I'll just read them out to Professor. So what we'll do is we'll take few questions as they come, and the rest, if I get any on my email, I'll compile them and I'll forward them to you.
Yeah, that's fine.
So participants, anyone with questions, please write them down in the chat box. You just wait a minute or so, otherwise I'll just take the questions on the email and compile them and send them to you, and I can individually. Okay, there's one from Tayyaba Idris. How does high heat tolerance of E. coli strains make them model organisms?
Excuse me?
Yes, there's one question from Ms. Tayyaba.
Yeah.
How does high heat tolerance of E. coli strains make them model organisms?
It does not make them model organisms, but the model organisms have acquired actually a new functionality of physiology. We don't know the impact on human health yet on such type of novel acquired features. However, one can imagine that in susceptible individuals that acquire heat tolerant Escherichia coli strain, there might be a higher risk of translocation, for example.
Dr. Samia Taslim has asked about your research group as a postdoc supervisor. What are the process?
We welcome applications, of course, but our possibilities are limited and we have to discuss this individually.
Mr. Zaidullah has asked, are there any disadvantages of E. coli using as a model organism?
Well, E. coli is not the only bacterium that is around on the planet. It is, of course, a very important human pathogen, and it's a very important commensal. That is, the importance is, I think, shown that in that way that it is one of the first colonizers of the human gastrointestinal tract, and it basically prepares for other organisms, in naturally born babies at least. And we know that, for example, babies born by cesarean section, they have another microflora, and also they are prone to have a higher susceptibility to certain type of allergies.
Mr. Ghulam Murtaza has two questions. The first one is, how can we recognize generally E. coli types? The other one is, E. coli moves towards stability over a large range of temperature. Then how can we sterilize or remove from specific systems?
Can you please repeat the first part
Sure,
of the question?
Sure. The first one is how we can recognize generally E. coli types.
There is a very simple, actually, PCR approach in order to recognize E. coli types. That is based on the presence of certain genes in the E. coli genome, but it can very effectively make a first classification into the phylotypes that I have shown on the phylogenetic tree. Sorry. And the classification into phylotypes already gives an impression whether, or a hint, whether this E. coli strain is actually a commensal. Commensal strains are often phylogroup A, while strains associated with persistence and or with virulence are phylotype B or other phylotypes. Then, of course, we have the classic microbiological techniques in order to identify Escherichia coli. And in these days, whole genome sequencing is very affordable and can characterize Escherichia coli strains on the single nucleotide level.
The other part of his question was that E. coli moves towards stability over a large range of temperature. So how can we sterilize or remove from specific systems?
Still, the standard sterilization techniques. Sorry. Still, the standard sterilization techniques are sufficient to usually remove Escherichia coli. It is also fortunate that the transmissible locus of stress tolerance is primarily present in non-pathogenic E. coli strains. Pathogenic E. coli strains have, at least at this time point, still a genetic barrier that does not allow the acquisition of the transmissible locus of stress tolerance. And that might be very fortunate, because if virulent strains have a higher tolerance against lethal heat stress, that might be a major threat for human health, equally as antimicrobial resistance is.
Hifsa Manzoor has asked why E. coli is used as an indicator organism to examine water or food is safe for consumption or not.
Because it is present in the gastrointestinal tract of almost every human being and is shed with the feces into the environment, which indicates a contamination. And E. coli can still survive, at least for a for a specific, for a certain time point in the water. So it gives a temporal indication for contamination.
Right. So I think those are all the questions. Any other questions, they can be dropped to me by email. The email is mentioned in the chat, and then I'll forward them to Professor Römling. With that, I would like to thank you, Professor Ute Römling, for your time and for such a brilliant lecture here today, and the over 100 participants that have joined us from various countries. Thank you so much for this lecture. Any other questions or details that I get, I'll compile them and then I'll forward them to you to answer.
Thank you very much for the invitation. I felt very honored to give this lecture today.
Thank you so much, ma'am.